Hi there. In the last two videos, I discussed ribosomal RNA genes and ribosomal RNAs. In this video, I'll go further talking about other types of genes, including transfer RNA genes and tRNAs, messenger RNA genes and mRNAs, and then small RNA genes and sRNAs. So first I'll talk about tRNAs and the tRNA genes. As I discussed in one of the previous videos, tRNAs all have this same general cloverleaf shape, where there's an acceptor arm up on the top, which has both the 5' and the 3' ends, including the CCA at the 3' end that's added post-transcriptionally. And then the D domain, which usually has a dihydrouridine within it, which is how it got its name D. And then the anticodon loop, which does contain the anticodon that attaches to the codon of the mRNA. A variable region, which is sometimes long, sometimes very short. The T domain, which often has a thymidine, as well as a pseudouridine within it, so it's sometimes called the T pseudo-C domain as well. And as I stated in a previous video also, there are these long-range interactions between the D and the T domain, as well as the D domain and the variable region. And these are indicated by these lines where you have hydrogen bonding, bringing those regions together. In the three-dimensional structure over here, you can see where those domains exist in the tRNA. So there's the acceptor arm here with the CCA tail sticking out, and then the D and T domains are interacting, brought together, so that they can push out the anticodon in the correct conformation so that it will attach in the ribosome to the codon of the mRNA. Here's a slightly different version of that, highlighting some of the key regions of the tRNA. So here's the CCA tail where the amino acid attaches. This is the D domain, and one of those nucleotides often is a dihydrouridine. And then the anticodon loop, often there's a U at the end and an A on the other side, and sometimes those are interacting. A variable loop, and then a T pseudo-C loop, or a T domain here, where you do have a thymidine within the RNA, which is normally found just in DNA, but it's found in RNA in this case. And this is the general shape showing where the amino acid would be attached to the tRNA and the anticodon exposure down here. As I indicated in ribosomal RNA genes, the promoter actually is part of the transcript, whereas in the mRNA genes, that's not the case. Here in tRNA genes and also the small ribosomal RNA gene, the promoters are actually within the transcript, further within the transcript, in the parts that become part of the mature tRNA. So in the case of tRNA, there's an A box and a B box, and transcription factor 3C recognizes those, attaches to them, and then transcription factor 3B can attach to that region, and that positions RNA polymerase 3 on the beginning of the gene, and then you get transcription of the tRNA. In eukaryotes, RNA polymerase 3 also transcribes the 5S ribosomal RNA genes. In bacteria, it's just RNA polymerase that does the job. But in eukaryotes also, the part of the promoter is within the gene again, and in this case there's a C box which attaches to transcription factor 3A, that then attaches to transcription factor 3C, but there are no A and B boxes, and then that attaches the transcription factor 3b, which then positions the RNA polymerase 3 at the beginning of the gene to start transcription. It's a little more complex than that in that for transcription of a tRNA gene, here's the A box and the B box down here. Here's transcription factor 3C, which you can see consists of 6 protein subunits. And the numbers in there stand for kilodalton. So there's a 90 kilodalton protein and a 138 kilodalton protein. And the 138 sticks to the B region, and the 95 over here, 95 kilodalton protein attaches to the A box. The 131 kilodalton protein down here attaches to transcription factor 3b, which also includes a TBP, which is a TATA binding protein. Now, in the case of these genes, there is usually no TATA region, which is usually a TATA or TAATA, something like that. Found in mRNA genes, the TATA binding protein attaches there. In this case, you still have the TATA binding protein. There's no TATA site. What you're getting is attachment of transcription factor 3b to transcription factor 3c, and then both of those attach RNA polymerase 3. So then RNA polymerase 3 is then positioned on the gene to start transcription at the +1 nucleotide. You can see the number of subunit proteins that are involved in producing our RNA polymerase 3 here. The 128 kilodalton protein, the 160 kilodalton protein. Many of these central proteins are in common with RNA pol 1, RNA pol 2, RNA pol 3, RNA polymerase in bacteria, etc. So the subunits added after that determine the specificity for the ribosomal polymerases, RNA polymerases in eukaryotes. But the basic central structure of that RNA polymerase is the same no matter what RNA polymerase you're talking about. So here's a basic phylogram showing this relationship between the eubacterial RNA polymerase up here, which contains only 5 subunits, and you can see them numbered here. And then you go up a little further on this tree, and more subunits are added. Between 11 and 13 subunits are present in the archaea, different groups of archaea. And then you go further up on this line, and then you have RNA polymerase 1, which has 14 subunits, RNA polymerase 2, which has 12 subunits, and RNA polymerase 3, which has 17 subunits, which is more specific, and it also transcribes a number of different small RNAs, including tRNAs and 5S RNAs, but also some others. So again, there are some minor changes in those central subunits, 1, 2, 3, 4, and 5. But in general, they remain fairly consistent throughout all of these, and they're performing the same function, right? They're doing the transcription itself. So those central portions are doing the transcription. The other proteins on there lead to attaching to different types of genes and transcription factors. So this is an artist's rendition of that, a little different view, where on the bottom here, these are for tRNA genes, where you first have attachment of transcription factor 3C, and then that brings in transcription factor 3B, and that then attaches to RNA polymerase 3, which is then ready for transcription starting at the +1 site. Up here with the 5S genes, then transcription factor 3A binds first, and then transcription factor 3C, and then 3B, and then finally RNA polymerase 3 to begin transcription of that gene. Some of the genes in eukaryotes occur as operons. And so in this case, only one promoter region is needed, and then an entire transcript is produced starting at that +1 position on the first tRNA. In producing that long transcript, it also produces several other tRNAs in the process. Then in the nucleus, there is processing of those, usually by RNA molecules, enzymatic RNAs, which separate each of the tRNAs plus a little more. So there are approximately 300 nucleotides in length for each one of those. Then there's processing, folding, and addition of the CCA tail. So then you wind up with all of these tRNAs being between about 70 to 80 nucleotides each. So again, those internal tRNA genes don't require those binding sites for the transcription factors. Only that initial tRNA on the 5' side needs to have those binding sites for the transcription factors, the A and the B sites within the gene. While most people think that the tRNA is what helps to decode the mRNA message, and it does, the code is actually set by a series of enzymes, the amino-acyl tRNA synthetases. So you have a tRNA for a specific amino acid code, so that's XXX for the triplet anticodon. And then you have the amino acid that should be put onto that tRNA. The thing that puts the amino acids onto the tRNAs, on those CCA ends, are these amino-acyl tRNA synthetases, and they are specific, or nearly specific. There are a few that aren't as specific, but specific for the tRNA and the amino acid. So they have sites on the protein for the tRNA and for the amino acid, and they attach them together so that you have the amino acid on the correct tRNA. And then it's called a tRNA that's charged with its amino acid. So the specificity of the genetic code is actually set by these protein enzymes, these amino-acyl tRNA synthetases, that add these specific amino acids to those tRNAs. It's been hypothesized too that originally some RNAs actually performed this function very early, 4 billion years ago. But then the tRNA synthetases that were proteins then took over this task, because they could be produced again in higher fidelity, and that they made fewer mistakes than did the RNAs, which were prone to mutation. So again, these amino-acyl tRNA synthetases set the genetic code, the specificity of the genetic code. And I will discuss these in a subsequent video in a little more detail than this. But I wanted to introduce this information prior to that. So this is just an introduction to show you that these AA tRNA synthetases are very important in setting the genetic code. So as I described in ribosomal RNA genes, I wanted to do this for tRNA genes, indicating the possible effect of mutations in certain locations of those genes. So you can have a mutation in the promoter that can increase or decrease the expression. But because the promoter is within those A and B sites, it can also affect the shape of the tRNA. Regulatory regions such as A and B can also, as well as other regions that attach the other transcription factors, those can also change expression levels. Sites for recognition of the amino-acyl tRNA synthetases, you could get alteration of the genetic code, which could be lethal to those organisms. There are introns in many tRNAs, and so if you have mutations in the intron, often that doesn't affect the tRNA much at all, or if any. If it's right on the borders of where the splicing sites are, however, it can be lethal for that tRNA and probably for the organism, or possibly for the organism. Again, if there are multiple tRNAs for the same amino acid, it may not have much of an effect to knock out one tRNA. But if in the case of bacteria where they may have only one copy of that particular tRNA, then it could be lethal. If you have a change in the anticodon, a mutation in the anticodon, that could alter the genetic code, and this probably it's one way that the genetic code did evolve, by having mutations in the anticodon or anticodon loop. If there are mutations that affect the structure of the tRNA such that it doesn't fit as well into the ribosome, then there could be lethal effects there. Although some mutations in the tRNAs that change the shape slightly have no apparent effects. And then if there are mutations in the non-control and non-structural regions, then there are no apparent effects. So again, as I said, for ribosomal RNA genes, please think about the selective pressures on the different tRNAs, and including in bacteria, where you essentially have one type of tRNA for each amino acid, and in eukaryotes, where you have multiple tRNAs for the same amino acid. And so there are some differences in selective pressures there. And these would be pressures in the nucleus, in the cytosol, in the cells themselves, and if they're multicellular, in the organisms. Because in multicellular organisms, which I haven't gotten to yet, there are different sets of tRNAs that are turned on and off, depending on whether it's a liver cell or a lung cell or an epithelial cell. You'll have different groups of tRNA genes turned on or off in those particular cells and tissues. So next are mRNA genes, messenger RNA genes, and these are the ones that everyone usually talks about first before talking about any of the other RNA genes. But I focus on ribosomal RNAs and tRNA, so I will go through mRNAs in a little less detail, because usually people have had many classes where they've talked about mRNA genes. In bacteria, many of the genes are included in an operon. So they will encode for more than one protein. In this case, the LAC operon is illustrated. There are two promoter regions, one for the LAC-I gene, and then another promoter here for the LAC-Z, Y, and A genes, all together as one transcript. So the LAC-I gene is here, it's transcribed, and it's then translated into a repressor protein. So when you have high levels of lactose, then that repressor protein is inactivated. The lactose sticks to it, and it won't stick to the promoter region for the other transcript, the ZYA transcript. It attaches to a specific region called the operator, which blocks RNA polymerase from binding to that region. It actually forms a loop in the DNA. So when there's lactose around, then this gene is turned on. And so RNA polymerase sits here and makes one long transcript. And so that makes, on the transcript, the mRNA, there's a Shine-Delgarno sequence that sticks to the anti-Shine-Delgarno sequence on the small subunit ribosomal RNA, which I discussed in a previous video. Then there's an AUG start codon, so methionine is added, and then translation continues to make this LAC-Z protein until it hits a stop codon. It's actually a stop codon, which ends translation for the LAC-Z protein. Then there's another Shine-Dalgarno sequence, which will again position this transcript on the small subunit of the ribosome. And then there's an AUG start codon for translation, and that's then decoded into the lac Y protein, and then there's a stop codon after that. And then another Shine-Dalgarno sequence, which will again attach to the small subunit ribosomal RNA and the small subunit of the ribosome. And then there's a UUG start codon in this case that makes the lac A protein, and then there's a stop codon at the end, a UAA. So the lac Z protein is the enzyme that takes lactose, which is a disaccharide, and breaks that into two monosaccharides. The other two proteins are ones that aid in bringing lactose into the cell. Once the lactose concentration drops, however, you don't need to make these proteins. So under low lactose conditions, the repressor protein then changes its conformation and will then stick to the operator region, and it will stop any transcription of that gene, which will stop production of the lac Z, Y, and A proteins. In the case of eukaryotic mRNA genes, they are usually single genes that make single transcripts that are then processed and eventually translated into proteins. Each gene, however, can make more than one protein if there are alternative splicing events that occur. And I'll talk about those in a later video. Right here, I've just made sort of a simple gene in this case. So often there are enhancers upstream from the gene, and those bring transcription factors and polymerase closer to the promoter region, so those enhance promotion of the gene. Then about minus 70 to minus 90 base pairs upstream from the transcription start site, there's often a GC region, high GC, and what's called a cat box, and those bind certain transcription factors. And then at minus 25, there's normally a TATA box, and I talked about the TATA box with regard to tRNA genes, which usually don't have a TATA box, but mRNA genes do have a TATA box, which does indeed bind a TATA binding protein, which is a transcription factor. So their transcription starts here at the +1 position, and there's a 5' untranslated region. That is, it won't be part of the message for the protein. And then there are exons and introns if the gene has introns. Some don't, but many do have introns. There's a second exon, a second intron, a third exon, and then a 3' untranslated region, which also contains a polyadenylation addition signal at the end. So the first thing that happens as this is being transcribed is that 7-methylguanosine cap is added, and then splicing occurs to bring the exons together and splice out the introns. And some introns actually have function later, so I'll talk about that as well. Poly-A tail is added to the end, varying in length from tens of adenosines to hundreds of them. Both the cap and the poly-A tail are needed in order for the mRNA to leave the nucleus, so those are both needed. The poly-A tail is important out in the cytosol because that controls to some extent the half-life of that mRNA, or how long it will survive in the cell. So there's a certain lifetime for each mRNA. And then you have translation after that, starting from the beginning of usually an AUG start codon and ending with a stop codon. So this is a little bit of the detail of what's going on to start transcription for an mRNA. So first you'll notice that the promoter region is outside of the transcript, so that it's on the 5' end, or 5' side of where transcription starts. And this is different from ribosomal RNA genes, where the promoter actually becomes part of the transcript, and in the case of tRNA genes, where the promoter is actually within the gene, part of the tRNA and part of the RNA that becomes the tRNA. So here the promoter is clearly outside of the part that's transcribed and translated. But you can see these enhancers upstream bind activators, and they're actually made from several different protein subunits. There are often silencers, which will repress the activity of the enhancer, so there's some control there as well. And then there are many, many other transcription factors, including the TATA binding protein and other transcription factors here, and then RNA polymerase. And as I've already shown you, RNA polymerase contains many subunits, from 5 to 17 subunits there also. So a lot of different proteins are involved in attaching the RNA polymerase to this site, activating it, and then beginning transcription of mRNA genes. So as I said, many of these genes have exons and introns, and I'll be talking about introns more specifically in a subsequent video. But just to show you some of the variation for eukaryotic genes, although a few bacterial genes also have introns as well, but they really have expanded in eukaryotes. So here's the globin gene, which contains 3 exons and 2 introns. And so the total length of the gene is 1,525 base pairs, but only 622 nucleotides or base pairs are within the exons, and the other 893 are within introns. The ovalbumin gene here has 8 short exons and 7 introns, so they only make up a small proportion of the total length of that gene. But I should first mention that the introns aren't just useless pieces of RNA. Many of them are used for other purposes, and some of the introns in specific genes, in some genes, actually contain other coding sequences, so they become mRNAs themselves, separate from the main mRNA that comes from these genes. The conalbumin, 17 short exons and 16 introns, and only about 20% of the gene is made up of exons. So just to show this visually anyway, here's the ovalbumin gene consisting of 7,700 base pairs of DNA. There is the 5' untranslated region. There's intron number 1, 2, 3, 4, up to 7 here, and then the exons, and then the final 3' untranslated region down at the end. So the first thing that happens is you get capping, poly-A tail added, and then splicing, and the spliced product, which will become the transcript for translation, is only 1,872 nucleotides in length, which could then produce a protein of over 600 amino acids in length. One of the extremes that I know of is the dystrophin gene, which when it goes bad causes muscular dystrophy because the protein doesn't attach part of the cytoskeleton to the membrane. So then the muscles can't move, the muscle cells. The gene is about 2.4 million base pairs in length, and the primary transcript is about 2.1 million base pairs or nucleotides in length. It takes about 16 hours to transcribe, so it takes a long time, but it has 79 exons and 78 introns. But the resulting mRNA that produces the protein is only 14,000 nucleotides in length. So more than 99% of the gene consists of introns, and less than 1% consists of the coding sequence for the protein. This one can be also alternatively spliced so that it can produce many different products, many different transcripts, and many different proteins, all isoforms. Once mRNAs are mature, that is, all of the introns have been chopped out and translation begins on a ribosome, this is the genetic code called the universal genetic code, but it's not quite universal in that several organelles and organisms use slightly different codes just in a few places. It's more or less universal. Something I'll be talking about later is the evolution of this genetic code. As you can see, these blocks in orange, these amino acids in orange, are nonpolar amino acids, and then the purple ones are polar amino acids. The ones that are green have a negative charge; those are acidic, and the ones that are in blue have a positive charge or are basic amino acids. So as you can see, there is a structure to this, and I'll be talking about this in greater detail in a subsequent video. But just to foreshadow that a bit, and I've talked a little bit about this in an earlier video, these ones on the lower row here are among the smallest amino acids. The 10 original amino acids found in the Miller-Urey experiments, as well as in meteorites, are found in this bottom row, as well as in these first two left-hand columns. So you can already see some structure and some possibilities as to how this genetic code began. And then up in this upper right-hand corner, those are among the most complex amino acids and some of the latest arrivals into the genetic code, and thus are found in more, modern proteins than in the ancient proteins. So in general, the aminoacyl tRNA synthetases put the amino acids onto the tRNAs, which have the anticodon, that then attach by hydrogen bonding to these codons, which are on the mRNA, and that's what delivers the tRNA with its amino acid to the ribosome in the correct position, and then that's added to the growing polypeptide chain to construct that protein. Another clue to the evolution of the genetic code is found here, where you look at the codon, the first, second, and third nucleotides in the codon with respect to what class of amino acid is being added. So nonpolar amino acids have usually a U in the center, although occasionally a C and G. The first and third nucleotides don't matter that much. So you can see that here with the U primarily, but occasionally a C or a G in that middle position. And then the polar amino acids usually have a U or an A in the first position of the codon and a C or an A in the second position of the codon. And you can see here that U and A are the most common in the first position, and the C or an A in the second position are the most common. Glutamine and serine here don't quite fit in to that, but I'll explain that later. Those were later arrivals to the genetic code. And all of the acidic amino acids, there's only two. They are G and A in the first and second positions of the codon, and anything in the third position. And here you can see them down here, a G in the first position, A in the second position, and the third position, anything, depending on whether it's aspartic acid or aspartate and glutamate. And that, again, is a big clue in the evolution of the genetic code. And then finally, the basic amino acids usually have a C or an A in the first position, an A or a G in the second position, and then anything in the third position. And you can see that here, a C or an A in the first position, an A or a G in the second position, and then anything in the third position. And you'll note from this that this third position can be quite variable. That actually is another clue to the evolution of the genetic code as well. So again, there are clues in this as far as the way it's arranged as to the evolution of the genetic code, and I will, go into this in detail in a subsequent video. So as with ribosomal RNA genes and tRNA genes, the position and type of mutation has many effects on whether that mRNA is transcribed, translated properly, etc. So if there's a mutation in the coding region, then that can make a mutant protein, but not always. If you have a mutation in the promoter, that can change expression, increase, decrease, abolish, whatever. The regulatory regions in there, as far as the enhancers and other regions, also can cause increases or decreases in expression. The regions on the 5' and 3' ends of the mRNA, which are non-coding, they're not translated, those can actually change transcription, they can change translation, they can affect mRNA stability, especially those signal regions on the 3' end that have to do with adding the poly A tail, as well as on the 5' end where the cap is added. That can change stability and whether the message is even translated, because it has to attach to the cap binding protein, which attaches to the small subunit of the ribosome. Introns, you can have altered splicing of exons that can affect both the ultimate protein that is produced and whether that's detrimental to the organism as well. And as I've indicated elsewhere, the introns, although they're spliced out, they're chopped out, some of those introns become small RNAs or controlling RNAs. There are some introns that have coding sequences within them, so they themselves become translated, and other proteins are produced off of those. So there are quite a few different types of things can happen with mutations in the introns. Interactions, if the mutations are within regions that interact with the ribosomal RNA, then translation can be altered. Interactions with tRNA, again, translation and decoding can be altered. Interactions with small RNAs also can control whether translation occurs at all for that mRNA. So those can affect anything from changes in splicing to changes in overall expression of that mRNA. And finally, what if the mutations occur in the non-coding, non-control regions of that gene, then many regions have no effect on the expression and the accurate expression of that mRNA and the mRNA genes. The final type of RNA that I want to talk about, these are non-coding small RNAs, and they have genes that produce them. Now, it's quite a variable group, so there are a lot of different small non-coding RNAs in a cell that do a wide variety of things in those cells. So this illustrates some of that diversity. So here's this gray ellipse here with RNA in the middle in white. So you have one branch here going to coding, which becomes the mRNA, right? So there's that one. All of these others, which are RNA, are non-coding. So there's the ribosomal RNA, there's the tRNA, which we've already talked about. And then there are these small RNAs that become snRNAs, which are small nuclear organizer RNAs that have to do with processing of ribosomal RNA as well as some other RNAs. And then there are several types that are included in this RNAi set, including Pee-wee RNAs, silencing RNAs, microRNAs. These all control expression of mRNAs primarily, of other types of RNA. So they can either hang on or attach to the mRNAs, thus restricting their translation, or they can cause these RNAs to be stored for a time, or they can cause degradation of those RNAs. So these are very important to controlling the mRNAs up here. If you look over here, non-coding RNAs, here's microRNAs, which will control reduced transcription, reduced translation, alternative splicing in general for some other small RNAs, X chromosome silencing over here, which is when females have 2 X chromosomes, only one is used, the other one is silenced. And that's silenced through attachment of the small RNAs to those sequences on the other X chromosome, which prohibits them from any type of expression of the gene. So only one set of the genes is on one chromosome is expressing at any one time. And the snRNAs do a couple of different things. They help to process the ribosomal RNA as well as some other RNAs, but they also cause nucleotide modification, so they can change nucleotides to some of those 120 different nucleotides that I spoke about in a previous video. So previously I talked about processing of ribosomal RNA. This one is a picture for yeast. And these processes of cutting the RNA and also modifying some of the bases is accomplished by snRNAs, those small nuclear organizer RNAs. So here's a figure showing one of the snRNAs, the U3 snRNA in red. And it has these different sequence boxes, the GAC box, the A box, the A prime box, etc. And then here's the primary transcript of ribosomal RNA. So the 5' end of that sticks to this region of the snRNA. That folds around, and then there's processing and clipping of that end, and then it proceeds down through box A and box A prime as well. And in folding that ITS1 here at this A2 site is then chopped. At the same time, this C, B, and D box, these down here, begin attaching to different parts of the ribosomal RNA, and then they will alter some of the bases specifically so that the ribosomal RNAs fold up in very specific ways. And this is a very ancient process, an RNA processing another RNA, something that first started over 3 billion years ago. Another group of these small RNAs are produced by a protein RNA complex called dicer, which will digest certain double-stranded RNA molecules. And it will cut them into these pieces that are approximately 20 or so nucleotides long. They then get attached to a RISC complex, an RNA-induced silencing complex, which uses one of those strands to bind to an mRNA, thus silencing it from any translation activity. So this occurs with both microRNA and silencing RNAs. So there are a couple different things can happen. For the silencing RNAs, which I was just describing, they will attach to the mRNA and signal it for digestion. So it takes it completely out of the translation machinery and won't allow expression of that mRNA. For microRNAs, on the other hand, they attach to a slightly different RISC complex and RNA, and those will attach to the mRNA and silence it, so it won't be available for translation. And in some cases, these are sequestered in certain regions of the cells called P-bodies. And so there's storage of large amounts of this RNA, this mRNA, for use later. So it's already been transcribed and processed, and then it's stored for quick action. So upon a signal, these can be released from those RISC elements and start to be expressed in large numbers in certain cells. So it's a way for a cell to rapidly respond to some outside signal. So this goes back to a figure I used in a previous video showing the process of transcription and translation. But then this highlights where those small RNAs are originating. So the small RNAs that control splicing here of heteronuclear RNA, and the SNO RNAs that control processing of ribosomal RNA, as well as the microRNAs, usually come out of processing of introns. So those are coming this way. Silencing RNAs here are usually transcribed by a different set of polymerases, RNA polymerases, RNA polymerase 4 and 5, and those are so those are coming from a different location in the nucleus. So at this point, you should go back over the small RNAs, tRNAs, ribosomal RNAs, mRNAs, and look at each region of each one of those genes that produces those RNAs. Think about the function of each region of both the gene and the RNA, and consider if mutations occur in different regions, what effect will those have on the functionality of the gene and of each one of those RNAs. And I have spoken specifically about some of those regions and less specifically about some of the other regions. We will cover some of the regions in more detail in subsequent lectures, but I think and videos, but I think that you can probably deduce on your own some of the effects, or at least a variety of effects that mutations would have in various regions of those genes that would be then transcribed into the RNAs, and what effect those would have on the functions of those RNAs. Okay, so that's the end of this section on tRNAs and tRNA genes, mRNAs and mRNA genes, and sRNAs and sRNA genes. So thanks for listening, and thanks for watching, and I will talk to you later.